Hello, welcome to ISMB 2024. My name is Dr. Marion Adebiyi, and my poster presentation is on homology modeling and molecular docking of some natural compound as inhibitors of Anopheles gambiae heat shock protein and bifunctional glutamyl prolyl transfer RNA synthetase. I'll be guided by the following outlines. Malaria is a disease caused by Plasmodium parasite and transmitted to humans from the bite of an infected female Anopheles mosquito. And despite the effort invested in malaria control, the World Health Organization reported 249 million cases and over 608,000 deaths in 2022. Figure 1 shows transmission and incidence of malaria as reported by the Center for Disease Control, where the endemic regions where malaria occurs throughout are represented in red. Then regions where malaria occurs in certain places are represented in yellow, and regions where transmission is not known to occur are represented in blue. Numerous strategies have been used over the years for the control of malaria vectors. However, there has been vast resistance that has been recorded, and this necessitated the need to find alternative compounds with insecticidal properties that will enable the control and management of malaria. Hence, one of our recent studies identified certain potential insecticidal target, which is the heat shock protein 70 kD and bifunctional glutamyl prolyl-tRNA synthesis. Additionally, some natural compounds from different plant species have been found to possess inhibitory effect on Anopheles gambiae. Hence, this study aims to model the 3D structure of Anopheles gambiae heat shock protein and the bifunctional glutamyl prolyl-tRNA synthesis, and to investigate the inhibitory effect of some of these natural compounds against Anopheles gambiae. We believe that this research will contribute to the development of novel insecticides for the control and management of malaria. In our research method, we employ homology modeling using Swiss model. Then after obtaining the model was refined and validated using Chiron web-based server and ProCheck to actually validate the 3D structure, after which we conducted molecular docking and post-docking analysis using the Pyrex software and Discovery Studio 2021 client. For the ATME tox analysis, we use Swiss ATME using the simplified molecular input line entry system specification for each of the compound, and these compounds were further analyzed using Orisys Property Explorer to assess the toxicity risks associated with each compound. Table 1 shows the global mean quality estimate of the proteins, the sequence identity, the sequence similarity, and the coverage, and we can see that most of the proteins, especially the Anopheles gambiae heat shock protein, had global mean quality estimate of 0.86. However, the Anopheles gambiae prolyl glutamyl heat shock tRNA synthesis had a lower global mean quality estimate, and this is due to the lesser coverage, since only 0.41% or 41% of the protein was covered during the analysis. Additionally, figure 2 represents the Ramachandran plot, and from the Ramachandran plot we have the Ramachandran plot statistics, where we have over 90% of the amino acid to be represented in the favored regions. Table 3A and Table 3B shows the binding affinity of the compound on each of the proteins. For the toxicity risk assessment of the compound, violacine had no mutagenic, tumorigenic, irritant, or reproductive effect. Likewise, sesamine, geldamicin, and cryophylline. However, ascaridol, halofuguine, pelitorin, and caviol had either mutagenic, tumorigenic, irritant, or reproductive effect. Figure 3 actually shows the 2D docked view complex of the top 5 compounds for Anopheles gambiae heat shock protein 70 kD. Figure 4 shows the docking interaction complex. That's the two-docked view complex of the top 5 compound for Anopheles gambiae prolyl-glutamyl-tRNA synthetase. And after these studies, we actually found that violacin had a significant binding and shows no reproductive irritant, mutagenic or carcinogenic effect, and this actually suggests for further validation of the role of this compound, which we believe would actually contribute to the development of novel insecticidal against Anopheles.